Hi, happy morning. Have a wonderful Wednesday. So, yesterday we have seen about Watson and Crick model of DNA, isn't it? Yes. And then I have posed a question in that. Then why the hydrogen bonds give the stableness to the DNA molecule? So, what is the meaning of stable? I asked you in the last session. Are you all with the answers? Yes. So the stable means the term stable means it's a steady, it's a firm. So the hydrogen bonds between the purines and pyrimidines give the stableness to the DNA molecule so that the DNA molecule will be in a firm condition. It will not be changed. It will be in a steady condition. Okay. So that's the meaning of the term stable. Yes. So now today we are going to see about the DNA replication. So, what do you mean by DNA replication? Any idea? The DNA replication means what? If anybody knows the answer, can comment. So, now let me see how many of you are coming with the answers for the DNA replication. Okay, so replication means what? Replica. The term replica means what? It's nothing but producing the same copy of the molecule. So here the DNA replication is the production or the synthesis of this exact copy of DNA. That is known as the DNA replication. Okay, so before understanding the concept in detail, okay, we should know what is the process which takes place in the cell. First and foremost process which takes place in the cell is the replication process. For any cell to divide, for the living cells, okay, for any cells to divide, they need the first and foremost step is the replication process. The DNA has to synthesize first. Then only the further RNA molecule or the protein molecule will be synthesized, isn't it? So now we can say that a DNA replication is a basic process in a cell. Is a basic process in a cell, we can say. Okay. So and then I told you that replication means the exact copies of DNA will be produced. The same copy of the DNA will be produced. That's the term replication is given. So it's known as the replica. Okay, DNA is going to synthesize its own copy of DNA molecule. A parent is going to divide and is going to give a daughter's new DNA molecule. That's known as the replication, a replica of the DNA. Understood. And then it is nothing but the nucleotides provide information to the new strand. What are the nucleotides? So the nucleotides are, what are the nucleotides? It is the substance which contain ribose sugar and it contains the nitrogenous bases, and along with that it will have the phosphate group. That is the nucleotide. So this strand, the nucleotide strand, is going to give rise to information to the new strand which is going to be synthesized. Is that clear? And then the daughter cells know that that is the daughter cells from the parent one. They will contain exactly the same genetic information. That is the replication introduction. Clear? So these are the introduction to replication. So the first point is replication is a basic process in all the living cells, and then during this process what will happen, the exact copies of DNA will be produced, and then the nucleotides provide information to new strand. Yes, and then the daughter cells contain exactly the same genetic information. So this is the general introduction to the DNA replication process. Am I clear? Okay. So let me explain the replication process an outline, in an outline form to you, so that before understanding the entire process, you will know the steps, the outline of the DNA replication. Is that clear? Now, see, this is the replication. What are the steps involved during this process? So the first step, what the DNA will do means, it will select the place as a specific site where this replication has to occur. Yes, so that is the first thing. So that is the origin of replication. So the first step, initial step, it has to choose the position or the specific point for the replication process. And the second thing is, it has to do unwinding. So without unwinding the DNA molecule, it cannot produce the new daughter DNA strand. So unwinding means what? Separation. The helical structure, no, the double helical structure of DNA has to separate, then only it can produce the another copy of DNA. So that is the unwinding of DNA. And the next thing is, it require one molecule for this formation. That is the RNA primer. What do you mean by RNA? Have you heard about RNA? I told you in the first live session itself, like DNA, there is another RNA is there, that is known as the ribonucleic acid. Do you remember that? So the ribonucleic acid will have the oxygen atom in its second position in the pentose sugar, whereas the DNA molecule will not have the oxygen atom in the second position of the pentose sugar. So this RNA primer, no, it will be formed at the site of the replication position, so that it will initiate the process. The nucleotides will be added by another enzyme. Okay, so that the RNA primer will trigger the replication process. So that there should be a formation of RNA primer during the replication. Understood. And the next is, there will be the synthesis of new complementary strand from the parent strand. This is the fourth step. So you know that complementary, what is complementary? If adenine is there, thymine will come and join. If cytosine is there, the guanine will come and join. That is the new complementary strand from the parent strand. So this is the general outline of replication. Understood? So first thing is origin of replication, the specific point where the replication should start, and the second thing is unwinding of DNA, that is the separation of DNA molecules, so that each strand will give rise to another double strand of DNA. And the next is, there will be the formation of RNA primer. This RNA primer only will initiate the replication process, and finally you will get the new complementary strand from the parent strand. Understood? This is the general outline of replication. Is that clear? Yes, I understood that you people are clear with up to this. Let me go in the next step. Okay, so first we will see about the replication, that is the origin of replication. Okay, I told you that it is the site of origin. What is replication? It is the site of origin. Origin of replication, nothing but the site of origin, and there will be a formation called replication fork will be formed. This is 2 points you should remember during the origin of replication. Is that clear? So look at this picture. You can understand better if you look into this picture. So this is the parent DNA. Okay, it is the double helical structure. It is going to find a position where it is going to start the exact copy of replication production. So now this is the place of origin, that is the site of origin where the DNA replication fork will be formed. So this red color circle which I have given here, no, this is the replication fork. Understood? So now This is the first and foremost step in the replication process. That is, the parent DNA is going to find a position, that is the site of origin, and there will be a replication fork will be formed. Thereby, the double strand of DNA, parent DNA molecule, will unwind. Okay, and then it will unwind and it will start producing the new daughter DNA cells. Is that clear? Now, next step is, what is the next step we have seen in the outline? We have seen that unwinding of DNA will take place. I told you, no, this is the unwinding of DNA. So here in this process there are some enzymes. What do you mean by enzymes? Do you have any idea about the enzymes? Enzymes are nothing but they are also the protein substances. They enhance the reaction. They enhances the any reaction, whether the chemical reaction which takes place in the living cell or it takes place in vitro. In vitro means in chemical labs also they will use some enzymes for to trigger their reaction, to enhance their reaction. So they will not take part in the reaction, but instead they will speed up the reaction. That is the enzyme's role. Okay, so here during the unwinding of DNA molecule, unwinding means what? During the separation of the parent DNA, there are two enzymes going to come and play the role. They are the helicase and the topoisomerase. Is that clear? So this helicase and topoisomerase know what they will do. So let me explain one by one. The first one is helicase. So this, what is the role of this helicase? The helicase, no, they will separate the two strands of DNA. They will separate the two strands of DNA. So look at this. This is the DNA double helical molecule, and there is a place site of origin has been selected. And then what will happen? Slowly the nucleotides, no, they will separate from each other, and then the bond between them will be separated. The hydrogen bond, no, is the main stableness of the DNA molecule, I told you, no. So this bond will be separated and the two helical structure will be unwinded. That is, they will be separated from each other from the helical structure. So this role was done by the enzyme called the helicase. Okay, and then next enzyme, no, that is the topoisomerase. I told you this topoisomerase, what they will do, they will separate the double helix and they remove the twist during the unwinding process. So when it is going to unwind, no, there will be a twist. That is, we already saw that helical structure, no, there will be a twist in between them. So that twist will be separated by the topoisomerase. So the topoisomerase will separate the double helix and they will remove the twist during the separation of the strands. Is that clear? So if you see here, I have chosen that this is the replication fork, and here the topoisomerase will come and play the role after the helicase. And then this separated DNA strand, no, this parent strand, they will be splitted into two, and that parent strand is known as the template DNA. Understood? So this template DNA only going to form the new nucleotide which is complementary to the nucleotides in the parent DNA, and it will form two strands. That is, the template along with the new strand will be produced. They will form two daughter DNA double strand. Understood? So this is about the unwinding of DNA. So what are the enzymes present there? Helicase and the topoisomerase. This helicase will do the separation of the two strands of DNA. That is, the helicase will separate the two strands. Only that job it will do. But the topoisomerase will separate the double helix, and the twist also will be removed. Along with that, the twist between the two helix also will be removed during the separation process. And then you will get the template DNA formation. That is a single strand of parent DNA. Template DNA is nothing but the single strand of parent DNA. So this is the second step of DNA. Understood? Clear? Unwinding of DNA. Now we will move to the third step. What's the third step? I told you in the beginning itself, in the outline process, there will be the formation of RNA primer. There will be a formation of RNA primer. Okay. So, this formation of RNA primer, what it will do? What is its role? It's mainly synthesized. This RNA primer, no? How it will become? It will come on its own. No. The DNA template strand I told you, no? That template strand only will synthesize the RNA primer. So the DNA is going to produce its own copy of DNA. Along with that, it is also producing a primer known as an initiation complex known as the primer. That is the RNA molecule. That is also produced by the DNA itself. It's clear. It is having its own synthesis process. Along with that, it is also producing the another molecule of that is RNA primer, which is also essential for the DNA replication. Okay, so this primer was also produced by the template strand at the replication site. Okay, the next is the short segment of RNA nucleotides. So this is nothing but the RNA primer are nothing but the short segments of nucleotides. In that, what is the difference? Instead of that is adenine will form a bond with a hydrogen bond, that is hydrogen bond with the thymine, no? Whereas in RNA, adenine will form a bond with the uracil. Okay, so that's the difference between DNA and RNA. Okay, so these things you will learn in your higher classes. What is the difference between the DNA and RNA? So now look at this picture. This is the RNA primer, and this RNA primer is nothing but the nucleotide which is produced by the template strand. The template strand is nothing but the single strand of the parent DNA molecule. Is that clear? So what is the formation of RNA primer? The RNA primer is also a nucleotide produced by the DNA template which is close to the replication site, and then this RNA, this is a short segment of nucleotide that will start the replication process. Understood? Once this RNA primer come and occupy the place, then the two strands, no, the two parent strands, they will start producing the new strand of DNA with the help of other enzymes. So let me explain the other enzymes. What are the other enzymes which are involved in the formation of new complementary strand to the parent strand? So what is the fourth step? What is the fourth step? The fourth step is nothing but the synthesis of complementary strand from the parent strand. So how it will be produced? The DNA polymerase is the enzyme. What is that? It is the DNA polymerase enzyme. What is the enzyme will do? It will add the new nucleotides to the formation of new strands. Understood? So for example, if it is A is there, then it will be thymine will be formed in the new strand. If it is C is there, G will be formed. If it is again A is there, T will be formed. If again C is there, G will be formed. So this is the this is a template, and this is the new complementary strand. Is that clear? New complementary strand will be formed like this only. So this is the adenine will form a bond with the thymine, that is double bond. Then again adenine, double bond. Then this one, this there will be a triple bond. I told you, no, in the during the study of structure of DNA, I explained to you about how the hydrogen bonds are formed. There will be double hydrogen bond between adenine and thymine, and then cytosine and guanine triple bond, and adenine and thymine double bond, and then cytosine and guanine triple bond. So this is the that is the top nucleotides are known as the template strand, and then below that this new strand will be formed by the new nucleotides which are complementary to the parent strand. The template strand is known as the parent strand. Is that clear? So like this, the new strand will be formed. So what is that? It is will be formed in a continuous manner. Yes, so the continuous one is the leading strand, whereas the short segment will be formed. So all of a sudden the DNA can't produce the continuous strand, no. So one strand will be continuous. One strand means what? One nucleotide strand will be continuous, and then another nucleotide strand will be a discontinued manner. That's a break in between them. The short segments will be produced. That will be a fragment formation. So that fragment formation is called the Okazaki fragment. Why this is called Okazaki? Any idea? So now today's this home assignment is to go and find out why it is called Okazaki fragment. Okay, this is the today's assignment for you people. Okay, so coming back to the fourth step in this, again and another enzyme DNA ligase will come and play the role. What it will do? Ligase means what? The enzyme which will join the short segments or the fragments. I told you one strand will be continuous, that is the leading strand. The another strand will be the short segments, that's known as the Okazaki fragments. So these fragments, no, they should be continuous, no, then only they can form a new complementary DNA strand, daughter strand, which should be double helical. How they will be double helical? Only if the strands are continuous. So for that they need another enzyme known as the DNA ligase. So this DNA ligase is an enzyme that is going to join the kuti fragments, that is the short segments, which is the lagging strand. So they will join them. So finally you will get this DNA, this is the parent DNA molecule. Then there will be the replication fork will be formed there. Yes, and then this replication fork, how it is formed? By the helicase and topoisomerase, and then there will be the separation of the two double strands and the unwinding of the twist of the parent strand, and then there will be the RNA primer will be there to initiate the new nucleotide formation, which is complementary to the template strand. Yes, so this is the template, that is the parent strand. Okay, this is the template and this is known as the parent DNA. Okay, and what is this? This is the RNA primer. Okay, this is the RNA primer. It is also synthesized by the parent DNA only. Clear? And then what will happen? This the RNA primer, it will initiate the process and slowly the complementary strand will be produced. So this is the complementary strand. Okay, so there will be that is the purines and pyrimidines, they will start bonding with the formation of new strands. See here, and like this they will get again this parent and this new strand, they will join and they will form the helix again, and in one stage they will get separated from the parent template. So this like this. Have you heard about the binary fission of amoeba? Yeah, so the same way the parent DNA molecule will get separated from the daughter nucleotide. So you will get two daughter DNA fragment. Is that clear? So this like this only DNA replication process takes place, and the DNA will produce the exact copy of its own. So that's known as the replication. Understood? Is that clear? So the fourth step is synthesis of complementary strand from the parent strand. So what is the enzyme playing the role? Is the polymerase enzyme. DNA polymerase only, it will add the new nucleotides so that it will get the new strand which is complementary to the parent. Is that clear? And then there will be the continuous production of the strand, that is known as the leading strand, and then the short segments are known as the lagging strands. So these lagging strands are called Okazaki Why they are Okazaki? That's a home assignment for you people to go and find out what is the Okazaki. And then this DNA ligase, it will join the short fragments and it will give the continuous strand formation. So this is the way the replication process will take place. Hope you understood very well the concept. Yes, now we will recap. That's a recap for you people again. What is the replication? How the replication takes place? Yes, shall I? Okay. Now, this is the replication process. What is the first step in replication process? Replication of, or that is origin. It's the replication of origin. That is origin of replication specific site. There only the replication starts. And the next is separation of the parent DNA molecule. That is the unwinding of DNA. And the next is formation of RNA primer. Understood? This RNA primer also synthesized by the DNA itself. It is also a nucleotide. It is ribonucleic acid. Okay, whereas DNA is deoxyribonucleic acid. Understood? And then last step is synthesis of new complementary strand from the parent strand. Is that clear? And then first step is, I told you, it is the origin of replication, where there will be the site of origin and the replication fork will be formed. What will happen? The parent DNA, it will form a replication fork like this. Understood? There will be the site of origin will be there and the replication fork will be formed. There only the parent DNA will untwist, will unmount, and it will get separated, and the parent DNA molecule is known as the template DNA molecule. Understood? That's the origin of replication. And then the unwinding of DNA will take place with the help of helicase and the topoisomerase enzymes. These two enzymes only going to do the process. The first helicase enzyme is going to separate the two strands of the DNA, and then what will happen? The topoisomerase, it will separate the double helix, and then it will remove the twist between the double helix. Understood? These two enzymes play a major role during the helicase and the topoisomerase, and the parent DNA molecule will separate like this. That's known as the template DNA. Is that clear? Yes. And the next is the formation of RNA primer. So I told you that DNA itself will produce an RNA primer that is near the replication site, and then this short segment of RNA nucleotide, that is the RNA primer, it will go and it will be formed in one, that is the replication site. From there the replication, that the exact copies of the new copies of the DNA molecule will be produced. So next, if you see the synthesis of a new complementary strand from the parent strand, so that's the fourth step. What will happen? Here the enzyme again, the polymerase enzyme will start producing the new nucleotides to the new strand, which are complementary to the parent strand. I told you that this is the template strand, is the parent strand, and the complementary nucleotide will be produced, that is the new strand, which was done by the RNA polymerase. Understood? And then this RNA polymerase continuously it is producing this new strand. So that strand which is continuous is known as the leading strand, and then the short segments are known as the lagging strand, which is also known as the Okazaki fragment. Find out this Okazaki, why it is called Okazaki. And then this fragments, the lagging strands should be joined together. They'll produce DNA that is, they will produce a continuous new DNA molecule, that is the DNA ligase will join the fragments. Understood? So that's what about the DNA replication. Yes, can you define replication now? What is replication? Replication is the process in which the new DNA molecule will be produced. The exact copy of new DNA molecule will be produced. That is known as the replication process. Yes, so if you have any doubts, please comment me and please give your feedback so that it will be very useful for me to improve myself in the upcoming session. Please comment me. I request you people to comment me. Then only I can improve myself and I can give the best in my upcoming videos. Yes, thank you. We'll see you in the next session. Okay, bye.